Hello everyone, this is Dr. Vishal Trivedi from Department of Biosciences and Bioengineering, IIT Guwahati. And what we were discussing about the different properties of a living organisms. And in that context, so far what we have discussed about the classification of the different organisms, and then we subsequently understand how these organisms are evolved over the course of a time. And then we have also discussed about the structure of the prokaryotic as well as the eukaryotic cell. And in the previous module, we have discussed about the different types of biomolecules. So we discussed about the nucleic acid, we discussed about the carbohydrates, we discussed about the proteins, and then we also discussed about the lipids. Apart from understanding the structure and functions of these biomolecules, we have also understood the role of these biomolecules into the cellular activities. Now, once we have understood the biomolecules and their structure and functions, it is important for us to understand how these biomolecules are playing the role in different types of activities. And this is what we are going to discuss in the current modules, right? So what is the central dogma of life is that you are going to have the DNA dependent DNA synthesis, which is called as the replication. So we are going to understand about the replications. Then we can also discuss about the DNA dependent RNA synthesis, which is called as the transcription. And then we also can have the RNA dependent protein synthesis, and which is also called as the translations. So let's discuss the first process, and the first process is called as the replications. The replication or the DNA replication is the duplication or the resynthesis of the genomic content, and it is essential to maintain the life of an organism, right? DNA has to be precisely replicated to maintain the sequence identical to the parent DNA. It will protect the appearance of the potential mutation and resulting changed phenotype. In the current lecture, we will discuss salient feature of the replication and the underlying factors as well as the mechanism. The replication of a DNA fragment has to be performed keeping the following point into the consideration. What is the point? The replication machinery must be duplicate the whole fragment, which means it is actually going to replicate the whole genome of that particular organism. It's not like you can do the partial replications. You cannot do the partial replications. The replication must be free of errors, which means there should be no mutations. So there should be no mutation. That is the ideal conditions. Although we know that there could be a mutation when the DNA is replicating from one copy to another copy. And then the machinery must be amplify the fragment in a given time frame. For example, the replicons has to be complete within the doubling time of an organism, which means there is a time restriction for the machinery to replicate the DNA. For example, the bacteria is actually, or the E. coli is actually going to duplicate into the 20 minutes, right? It's going to make the one bacteria into two bacteria. This means the replication has to complete within this time frame. Now, before getting into the details of the DNA replication, it is important to recall what are the properties of a particular DNA. Remember, when we were talking about the nucleic acid, we were discussing about the different types of properties. So, what are the different properties? Just we can, very quickly summarize or review what we have discussed, right? So DNA has the 4 nitrogenous bases, right? We can have the 2 of the purine bases, where you have the adenine and guanine. Then you can have the 2 of the pyrimidine bases, which are the one ring bases. These are called as the cytosine and thymine. And these 4 bases are linked in a repeated pattern by the hydrogen bonding between the nitrogenous bases. You remember that the A is always making a double hydrogen bonding with the T, and G is always making a triple bond with the C. So the linking of the 2 of the complementary strand is called as the hybridizations, which means A is always going to make with the pair with T, and G is always going to make with the C. And this is important because that's how the DNA is actually going to maintain the complementarity in their sequences. So what is mean by the complementarity? Complementarity means is that you are going to have the primary sequence, right? So you can imagine that this is the first strand synthesis, and then this is going to be complementary, right? Because the A is actually going to make a pair with G and T, and the G is always going to make a pair with C. So wherever you have the G, it is going to be C, and wherever you have the A, it is actually going to be T. And that's why the sequence on the primary first strand is going to be the and on the other hand, it is going to be complementary strand, and that's why the first strand is called as the primary strand and the second strand is called as the complementary strand. So before getting into the detailed process of the DNA replications, let's first discuss about the machinery what is responsible for the replications. So we have the DNA replication machinery. So what is there in the replication machinery? The replication of the DNA required the following enzyme as well as the factors. The first enzyme what is required is called as primase. This enzyme synthesizes the primer at the site of replication to start the replications. So this is going to be the first enzyme which is going to synthesize the small stretch of RNA, and that small stretch of RNA is actually going to function as the primer. Then we have the DNA polymerase. These enzymes are responsible for the synthesis of the DNA of pre-existing fragments. So DNA polymerase is the main enzyme which is also called as the DNA dependent DNA polymerase, and these enzymes are incapable to perform the de novo synthesis but can be able to synthesize the DNA on a pre-existing RNA or the DNA primer. What it means is that DNA polymerase cannot synthesize the de novo synthesis. They actually can add the nucleotide on the existing DNA fragment, which means if you have a DNA fragment, the DNA polymerase will not be able to add the nucleotides onto the other strand. But if you have a small strand like this, then it actually can add the nucleotide on this according to the nucleotide what is present onto the primary strand. So this is going to be complementary strand, this is going to be the primary strand, and that's why this also require the action of the primase, because the primase is actually going to synthesize this RNA primer, and that's how this can be utilized by the DNA polymerase to synthesize the second strand. DNA polymerase can have the three different types of enzymatic activity. It can have the 5' to 3' polymerase activity, which means it actually going to do a DNA synthesis into the direction of 5' to 3'. Remember that when we were talking about the DNA, we were talking about that it has a 5' phosphate and the 3' hydroxyl group, and it is going to be complementary. So it's going to be like this, right? Which means on the 3' you are going to have the 5', and that's why you are going to add the 5' in. So this is going to be DNA synthesis. Then it also has the 5' to 3' exonuclease activity, which means that is going to degrade the DNA. So DNA degradation activity, and that activity is also nucleotide specific. And then you also have the 3' to 5' proofreading activity. So E. coli has 5 different polymerases, but only 3 are involved in replication and 2 are involved into the repair or to the recombination. So 3' to 5' proofreading activity is that it is actually going to check the errors into the DNA synthesis. So now you can have the 3 different types of DNA polymerases what is present into the organisms, and these DNA polymerases are called as DNA Pol 1, DNA Pol 2 and DNA Pol 3, that are going to be synthesized by a gene which is called as Pol A, Pol B, Pol C. Then it also has the different amount of subunits, right? So in a DNA Pol 1 you have the single subunit, whereas in the DNA Pol 2 you can have the 7 subunit, and in a DNA polymerase 3 you can have the more than 10 subunits. The molecular weight is also different, so you can have the 100 kDa, 88 kDa and the 790 kDa. Then the 3' to 5' exonuclease activity, so that is present in the 1 and so that is present in all the 3, right, 1, 2 and 3. Then you can have the 5' to 3' exonuclease activity, so that is present in 1 and 3, but it is absent in the DNA polymerase 2. Then you can have the polymerization rate, which means it is going to be 5' to 3' polymerization activities, and that is 16 to 20 nucleotides per second. In the case of DNA polymerase 1, where it is going to be 40 nucleotides and per second, and it is going to be 250 to 1000 nucleotide per second, which is for the DNA polymerase 3. Then the processivity is going to be 3 to 200 in the case of DNA polymerase 1, 1500 in the case of the DNA polymerase 2, and it is going to be more than 500,000 in the case of DNA polymerase 3. Apart from the DNA polymerase and the primase, you can also have the DNA gyrase. So this enzyme is required to unwind the double stranded DNA. Remember that when we were talking about the plasmid isolation, we said that you can actually be able to use the DNA gyrase to convert the different form of the enzyme. And then you also require the DNA ligase. So this enzyme is required for joining the DNA fragments. And then you also require the single stranded DNA binding proteins. So after the unbinding of the DNA by the DNA gyrase, the SSB is going to bind to the single stranded DNA to avoid the rebinding. It helps to reduce the energy required for the unbinding, which means once you have the double stranded DNA, and if you DNA gyrase is going to bind, it's actually going to make the single stranded, right? It is going to remove the unbinding. But once it removes the unbinding, since the DNA has the indigenous ability to rotate and it gives you the double stranded DNA, it actually, the single stranded binding protein is actually going to bind to the all the nucleotides, and that's how it is actually going to allow the DNA to be remain as the unbinded, right? And because of that, it actually can be utilized by the different components of the replication machinery, like for example the primase or the DNA polymerase, and that's how it is actually going to facilitate the DNA synthesis. Now let's discuss about the processes. So the processes is, first you have to identify the site of occurrence, like where the replication has to start, right? So it is difficult to start the replication of circular or very large DNA fragment randomly, right? You cannot start the work randomly, right? When you go for a site, right? For example, if I have to start making a building, right? I have to go for a site. I have to first inspect where I could have the front door, where I could have the back door, where I could have toilet, right? Those kind of treatment, right? So when the machinery will go to the site, it actually cannot start randomly. It has to first decide from which side I should start making the road, which side I should start making the different types of things. So hence a particular pattern of nucleotide sequence existed on DNA to recognize and facilitate the replication initiations. The difficult task with regard to replication is to denature the double stranded DNA, and the presence of sequence with low melting temperature will facilitate the event. So what is the most difficult part in the DNA replication is that conversion of a double stranded DNA into a single stranded DNA, right? Once you do that, then the other tasks are very easy, because then you can actually have the primase which is sitting and then synthesizing the primers, and then subsequently you can have the RNA polymerase, and that also going to add the nucleotides. So interaction between the adenine thymine is mediated by the 2 hydrogen bond, whereas the guanine in cytosine is mediated by the 3 hydrogen bond. So where you can actually do the melting, where you can actually do the denaturation easy, because the A is making 2 base pair with T and G is actually making the 3 hydrogen bonding with the C. So this is actually going to be stronger. So you can actually be able to look for the sequence where you can have the more amount of A and T, and that is actually going to say that okay, this is the site from where you can actually be able to open the DNA very easily. So as a result, the presence of AT rich region will facilitate the earlier denaturation and assembly of the replication machinery. These sequences are mostly present in a region and known as the origin of replication. So the portion where you're going to have the AT rich sequences, that is going to be called as the origin of replication. So you see this is a origin of replication of an E. coli. And what you see here is the different types of components, like you can have the 13 base pair tandem repeats, and you can also have the this sequence, right? So you see here it also has a very high quantity of the AT rich sequences, and these are actually going to be the site from where you can actually start the replications. Once you have identified the site of occurrence, then all other machinery is actually going to installed onto this particular sequence, and then it is actually going to start the replications. Then we can have the first event, and that is called as the initiation. So what you have here is, this is the initiation. So the unzipping of the DNA at the origin of replication forms a Y-shaped structure known as the replication fork, right? So once you have once you have the identifying the site, okay, then you are actually going to unzip the DNA, and once you unzip the DNA, you are actually going to form the Y-shaped DNA, right? And this Y-shaped DNA is called as the replication fork. And then what will happen, the first is that RNA primase or the primase is actually going to sit onto this small stretch of DNA which is going to be open, and then it is actually going to synthesize the RNA primer. Once the RNA primer is synthesized, then on this you are actually going to sit the DNA polymerase, and it is going to start doing the synthesis. You see, as the fork is opening by the molecules like the helicases as well as the topoisomerases. So topoisomerases are keep moving in the forward directions, right, and keep opening the DNA. And that's how this Y-shaped replication fork is going to be more and more available for the machinery to replicate. A short stretch of DNA is formed at the 5' end. This is the RNA primer, and it is synthesized by the primase. Synthesis of RNA primer is essential as the DNA polymerase cannot perform the de novo DNA synthesis. Now we can have the elongation. So elongation is nothing but the addition of the nucleotides onto the different incoming new 3' ends. Okay. So in the elongation step, the DNA polymerase start the synthesis of the DNA onto the pre-existing short oligonucleotide strands. In this step, an incoming deoxynucleotide gets joined by the hydrogen bonding to the appropriate nitrogen base of the single stranded chain as per the base pairing loom, which means A is making a pair with T, T is making a pair with A, C is making a pair with G, and the G is making a pair with C. The nucleotide triphosphate joined to each DNA strand break off their high energy bond and set the free pyrophosphate. So it's actually going to set the free pyrophosphate, which is actually going to digest, and it's actually going to generate the free phosphate. And in this process, it's actually going to have the energy regulations. So the pyrophosphate undergoes hydrolysis with the help of an enzyme which is called as the pyrophosphatase, and it releases the energy and set the inorganic phosphate to Pi. The energy released in this process is used to derive the polymerization of the nucleotides to form the DNA. The released deoxyribonucleic monophosphate joined to each single stranded DNA become linked to form the new DNA chains. Okay. Then we have the DNA polymerase. DNA polymerase can polymerize the deoxynucleotides in the direction of 5' to 3', which means it actually can synthesize on to the 5' to 3', right? As the DNA strands are anti-parallel to each other, the new strand must be formed onto the older strand in the opposite directions. The new strand forms continuously into the 5' to 3' directions. So this is the strand which actually is going to form as a continuously, like it's going to run along with the replication fork, where so this strand is called as the leading strand. So this strand is called as the leading strand, right, because it is actually going to synthesize along with the opening of the replication fork. So the strand which going to be go for synthesis in the same direction as the replication fork, that is called as the leading strand. Whereas on the other parent, on the other strand, like on this strand, it is actually going to wait because the DNA synthesis is always been in the 5' to 3' directions, and you know that you have a 5' direction, so it's going to be a 3' here. So this means it has to wait for the DNA to go for some unzipping, and then the primase is actually going to sit and synthesize the RNA primer, and then you are actually providing a 3' site, and that's how it is actually going to utilize and go for a synthesis of 5' to 3'. This means on this strand, on the other strand, the short DNA strands are synthesized into the 5' to 3', and these short stretches are known as the Okazaki fragment, and these fragments are joined to give together to give the lagging strand. This means this is going to be the leading strand, and on this one, this strand has to wait for the DNA to be unwind for some spot of time, right? So this is going to unwind, it's going to, RNA primase is going to sit and going to synthesize the primer, then this small stretch of DNA is going to be synthesized. Then it has to wait for some more time, then this amount of DNA is going to be open, and then again the RNA primase will sit, and then it is going to be synthesized. So these individual fragments are called as the Okazaki fragment, and since this strand is running in the opposite direction to the direction of the fork, this strand is called as the lagging strand. The RNA primer is replaced by the deoxynucleotide, and the gap is joined by the enzyme which is called as the DNA ligase. So eventually what will happen, these RNA primers are going to be removed, right, and then this is going to be filled by the deoxynucleotides, and the gap is going to be joined by an enzyme which is called as the DNA ligase. Now once the elongation is over, then it is going to have the termination. So once the replication fork is going to reach at the end of the DNA or meet the terminal region which contain the multiple copy of the TER sequences, the TER sequence are going to trap the halt of the replication, which means once the replication fork is going to be reached to the end of the tube or end of the DNA, then it is actually going to have the multiple TER sequences or the termination sequences, and these termination sequences are actually going to stop the movement of the replication fork, and that's how it is actually going to have the terminations. Apart from this particular type of activity, you can also have the proofreading as well as the DNA repair activity. So the template nucleotide sequence directs the accurate incorporation of the incoming template and ensure the accurate DNA replication. After every round of nucleotide incorporations, the DNA polymerase runs in the backward direction and check for the accuracy of the incorporation. If any errors is detected, it is corrected at this stage. So what will happen is, when the DNA is synthesizing, right, it is actually synthesizing in this direction, but the DNA polymerase is also moving into the reverse direction and keep checking whether the nucleotide what he has added is as per the template DNA or not, right? If it is not as per the template DNA, then it is actually going to remove those nucleotides by utilizing its exonuclease activity, and then it actually going to do the synthesis again. So that's why the DNA proofreading and the DNA repair activity is important for the successful synthesis of the DNA and making a copy which is almost identical to the host DNA. So now what we have discussed about the different types of steps, right? So what we have is, we have the 3 events, right? We can have the initiation, right? So in the initiation, what will happen is that the first is that it is actually going to recognize the origin of replication. So origin of replication, it is going to synthesize or recognize the origin of replication, and origin of replication is actually going to have AT rich area, right? So it's at the AT rich area, the helicases and the topoisomerases are going to go and sit, right? So they will actually going to have the double stranded DNA, right? And imagine that if this is the region where you have actually the AT rich area, so on this area the helicases and the topoisomerases will go and sit, and then they are actually going to open this strand, right? So they are going to open the middle area, right? And that's how this actually is going to form the replication fork. So once the replication fork is formed, right, so this is going to be formed, right, so then it is actually going to start the synthesis, right? So it's actually going to allow, so in the initiation, first is this, the second, the primase is going to sit, and then it is actually going to synthesize a small stretch of RNA, right? So it's going to synthesize the small stretch of RNA, right? So it's going to synthesize the small stretch of RNA. Now remember that this strand is actually a lagging strand, so on this side, and then it is actually going to have the third event. Third event is where you are actually going to have the DNA polymerase, and DNA polymerase is actually going to have the synthesis of the DNA. So DNA polymerase is going to sit and it is actually going to start the synthesis of the DNA. So it's going to start uh, synthesis of the new strand, right? So it's going to start synthesis of the new strand, and it is actually going to see what is the nucleotide present. So if the A is present, it is actually going to have the T, and if it is G is present, then it is going to have the C, and if the T is present, then it's also going to have the A, and if the C is present, then it is also going to have the G, right? And utilizing that, it actually going to reach to the end, right? So then the DNA replication is going to start, and for this strand it has to open for some time, and then from this side the the primase is actually going to sit, right? So primase is actually going to sit and start going to synthesize the small stretch of RNA, right? So it's going to start the synthesis of primer. So this is actually a primer, right, which is going to be synthesized in both the cases. And then the DNA polymerase is going to go and sit, right? So DNA polymerase is going to go and sit and start the synthesis of the DNA, right, in this reverse direction. And that's why it is actually going to have the multiple such fragments, and these fragments are called as the Okazaki fragments. And that's why this is going to be the leading strand and this is going to be the lagging strand. Now, once they reach to the end of the tube, so after this is going to be a second step, right? It's going to be elongation, right? So you're going to have the elongation, and the elongation is where the DNA polymerase is going to synthesize the DNA, and the third is you are going to have the termination. So once this DNA is actually going to reach to the ends, right, it's actually going to have the terse sequences. So these terse sequences are actually going to be rich in the GC sequences, and because of that it is actually going to stop the movement of the fork, right? Because see, the fork is moving in this direction, it's actually going to stop the stop the replication. So that's why this strand is going to be go and synthesize up to this, and this one also is going to be synthesized in the reverse directions. So this is all about the central dogma of life, and what we have discussed about the many events, what is happening inside the cell and what is responsible and what are different types of molecules are going to be produced. And what we have understood is that the protein is actually being responsible for making the different types of activities within the cell, and the protein synthesis is always being governed by the sequence dependent synthesis of the RNA, and the sequence dependent RNA synthesis is being done by the DNA. And all these events are actually being coming together under the central dogma of life, or to the central dogma of molecular biology, as it is being stated by the Francis. What we have discussed about the replications like the DNA dependent DNA synthesis, which is being done by the DNA polymerase, and we have discussed about the different events where we have discussed about the origin of replications, we have discussed about the initiation, elongation and terminations. So with this, I would like to conclude my lecture here. In our subsequent lecture, we are going to discuss some more aspect related to the central dogma of life, where we are going to discuss about the transcriptions. So with this, I would like to conclude my lecture here. Thank you.